At Mantle Biotech, we believe that everyone deserves access to high-quality medical diagnostics. That's why we're reinventing point-of-care assays, designing our tests to make them accessible to the patients who need them the most. To make tests that are more thermally stable, affordable, and accurate, we're producing biological reagents sourced from thermophilic bacteria found living in hot springs. And we're leveraging the most advanced techniques used in the pharmaceutical industry. We're Mantle Biotech, and we're harnessing extreme biology to take on unmet needs in global health.
Thank you. We're about to take a deep dive on the science. I would ask that you all put your nerd hats on, stick with me, because I promise you, and this is a money-back guarantee, this is the most excited you will ever be about urine-based diagnostic testing. The diagnostics industry has been stuck using the same fundamental technology for over 50 years. This comes in the form of this floppy molecule behind me, which is called an antibody. Antibody are binding proteins that grab onto other molecules, and each distinct type of antibody recognizes its own particular binding target with unique lock and key specificity. That's what makes these binding proteins useful in diagnostic tests. They allow us to recognize markers of disease floating around in complex, messy patient samples. You might have actually encountered a test just like this in the form of the common low-cost pregnancy test. We think that this is a really ideal form for diagnostic tests. It's accessible, it's immediately clear what it means. But unfortunately, it's not really adaptable to many other disease targets yet. In large part, that's because these antibodies aren't actually well suited for this sort of point-of-care assay for a number of reasons. One, they're slow to develop and costly. They're also expensive to produce, typically because you have to make them in animals. And they're relatively thermally unstable, which means if you're trying to make a point-of-care test that you're sending to rural clinics or you want it to sit on the shelf for 2 years, that's difficult. So we said, maybe this status quo just won't allow us to make diagnostic tests like this for all diseases. Maybe we need to get rid of this archetype and develop a new kind of binding protein altogether. So we said, how can we find a protein that addresses all of these limitations? Specifically, we want something that we can quickly produce in a test tube, something that we can make in bacteria, and something that is intrinsically thermally stable. So we said, let's look to an environment where proteins naturally would have evolved some of those characteristics. Let's look to that violent interface where the Earth's molten core contacts the watery biosphere. Let's look to hot springs. In particular, to a protein made by a microbe living in those hot springs called SSO7D. This protein can survive temperatures up to 98 degrees Celsius, so you can practically boil it and it will remain functional. It's like frying an egg, but the egg just won't fry. And what we like about this in particular is it has this really well-defined structure. So it has that thermally stable core, and that's what renders these proteins robust. And then it has a binding face that points outwards. That's the business end that interacts with other molecules floating around. And because it's structurally isolated, we can actually swap it in and out in this plug-and-play modular fashion. That means we can develop new kinds of binding proteins based on this common thermally stable scaffold without losing that thermal stability. We've now done so for 10 different diagnostic targets in our lab. And along the way, we found that we can make these proteins hundreds to thousands of times cheaper than traditional antibodies, and they're over 200 times more stable. What's more, our patent-pending development techniques allow us to rapidly iterate on that process, 2 to 4 times faster than the industry standard. That means that in the time that it takes you to produce a single antibody and a single test based on that antibody, we can rapidly produce many tests based on our binding proteins, both by iterating in a tube, developing new binding proteins, and also parallelizing those efforts using high-throughput robotics. We're first applying this platform technology in the realm of active tuberculosis, where 36% of all cases go undetected every year. And this is in large part because the diagnostics that are currently out there for TB, they're not well suited for it. They don't actually make it to the point of care. And so studies have shown that if you had a test that was affordable, that was accurate, and that was accessible, you could save up to 300,000 lives every year. We're well on our way to making that exact test, producing a paper-based assay that costs less than $1, and that we can actually validate in early 2020 in our clinical pilot. But that's just the tip of the iceberg. Active TB is a huge global problem, but there are many medical conditions that would benefit from this sort of point-of-care diagnostic test. We want to address each of these targets. In fact, we want to address the whole of the global immunoassay market, currently valued at $20 billion. And we think we can do that by rapidly iterating on this development process and expanding the market, basically meeting unmet needs within the medical industry. We think that we're the team to do it. My name is Eric Miller. I'm a recent graduate of the Chemical Engineering PhD program here at MIT, and this is the subject of my thesis work. Aditi Trahan is our clinical expert with backgrounds at the NIH Undiagnosed Disease Program and the Broad Institute here at MIT. And my PI, my advisor, Hadley Sykes, is an expert in protein engineering and medical diagnostics. We have significant traction in this space. We have 3 pending patents and a 4th that's disclosed, 7 peer-reviewed publications to show that it's real, and a research collaboration established with a Fortune 100 company that will allow us to actually make hundreds of millions of tests like this each year. Additionally, we've received the generous support of many partners and funders, and we're always looking for more. So if you're an interested investor, if you're an interested corporate partner, if you're a potential teammate who would like to help us to achieve this vision of making universally accessible diagnostic tests, please come find us at the booth afterwards. At our core, we are making diagnostic tests that are accessible, affordable, and accurate. We're Mantle Biotech, and we're leveraging extreme biology for extreme impact in human health. Thank you.